Hello students, so this one is another video on VIVA questions on the topic that is basic lab practices. Okay, so basic lab practices, you should follow them. You should know what are basic lab practices, and yes, they are important while you are studying microbiology. Okay, so already I had uploaded many videos on VIVA exam, okay, or say for your interviews also. So you will find some questions getting repeated, okay, in this video. So I just want to remind you that these are the questions which are likely to be thrown at you during your vivas, okay. Very simple logical questions, and if you're not prepared for these, then it is difficult for you to crack your viva. Okay, so you should be prepared for all these simple techniques and simple viva questions on them. Okay, so just to help you out for your viva exams, this is another video. Okay, so before starting to lab practices, we will go through some basic microbiology techniques. So what are basic microbiology techniques? Very basic, and first is inoculation, then streak plate, pour plate, spread plate, and sterilization and disinfection. These are very important basic techniques which are taught in your first year, right? So some questions on that. So you will think, what, I can get a question on inoculation. So yes, you will get a question on inoculation as well, like how to ensure that inoculation is done properly. How will you know that the inoculation that you had performed now is done properly? So you can answer it like this way, that by following aseptic techniques, preparing good culture suspension, or using a master plate inoculation, proper storage of cultures. These are the points which you can follow to ensure that your inoculation is done properly. Okay, so say if you're not following aseptic techniques, then you are not inoculating properly, you are actually contaminating your media. If good culture suspension is not prepared, that means you took or you pick out a very small portion of your colony, or you just touched your colony and you tried to make a culture suspension. So the number of bacteria which you are carrying from the main colony to your saline suspension is very less, and thus your culture suspension is very dilute. So the chances of getting the microbes or microbe or the culture being transferred or inoculated is also very less. Okay, the next is using a master plate inoculation for inoculation. Okay, so you use master plate for inoculation is you directly pick a colony, a pure colony from your master plate, and you inoculate in a broth. Okay, so that is one of the techniques that is followed for inoculation. And next is proper storage of culture. If you had isolated a bacterial colony, and you had made 2-3 master plates of that particular culture, but you have not stored your master plates properly, then say for 5 to 6 days those plates are lying outside of your refrigerator or incubator, then what will happen? Those cells will gradually die. Okay, so that also depends on the type of your culture, but I am just giving you a example here. Okay, an example here. So these are the points which you should follow to ensure that inoculation is done properly. The next is, does turbidity of your culture suspension ensures good inoculation? Okay, so the answer is yes. How? So a good turbid culture suspension, it ensures that a good number of microbial cells are getting transferred to your plate or your broth. Okay, so that is how you ensure that yes, my inoculation is good. One of the criteria. The next is, when to expect visible growth in liquid or solid media. So that depends on the type of microbe that you are using. Some microbes like actinomycetes, they are slow growers and they take more time than bacteria and fungi. So you should answer like, it depends on the type of microbe we use. For example, for bacteria, in 16 to 18 hours in liquid media, you can see turbidity. And in 18 to 22 hours on solid media plate, you can see small colonies which are popping up, okay, growing. In case of fungi, in 20 to 24 hours in liquid culture or in liquid media, cottony strands or balls are observed. Okay, you will observe cottony balls in your liquid media if the flask is kept on a rotary shaker. Okay, as the media keeps moving all the time, these strands they accumulate to into a ball. Okay, and in case of solid media, in 18 to 24 hours, you will find small colonies, soft cottony colonies of fungi growing. Then how to know if the instrument or equipment is calibrated or not? Okay, so you uh, walk into your uh, practical examination hall and you are about to weigh something on your analytical balance, and suppose the supervisor or the examiner walks towards you and he just simply asks, or he just throws a question to see what you are answering, that do you think this instrument is calibrated or not? Okay, so how will you check? So the simple answer to this is you first go towards logbook, okay, which is dedicated to that particular instrument. You check the entry if it is been calibrated in recent days, or you directly ask to the supervisor, okay, your lab attendant or your supervisor. Okay, so these are two simple ways that you can ensure that a given equipment or instrument is calibrated or not, right? The next is how to use how is used media or culture plates discarded. You should also know this. Many a times I have seen even my colleagues just using the given media plate, incubating it, recording results, and just keeping that plate beside the wash basin. And they don't even care to ask the supervisor or the lab attendant that what is done next to these used culture plates. So the answer to that is these samples. So samples, I mean here is used uh, petri plate or broth, okay, or any semi-solid agar that is been used, and it has any culture, okay, known or unknown culture. It is first treated with disinfectant, okay, for at least 12 to 18 hours or even 24 hours, and then the media is scrapped out. It is collected into a autoclavable bag, and then it is autoclaved and then incinerated. Okay, so why autoclaved? To ensure that all the bacteria or any microbes which are growing in that particular media, they are killed. Okay, and then only that used media is discarded. Okay, so it can be um, thrown into say wastewater, or it is given to a wastewater treatment or sewage, or say it is simply incinerated. So that depends on the area where your college is or your institute is and what method they follow. Then next is why to use saline solution for making cell suspension. Why not water? Okay, so another simple question from basic microbiology lab practices, that we don't make saline suspension, a culture suspension in water. We make it in a saline solution. Okay, so why saline solution and what is saline solution? Let me just remind you here. We take around 0.85 grams of NaCl, or sometimes even zero point 0.9 grams of NaCl is used in some colleges, and it is added to 100 ml of distilled water. Okay, and again it is autoclaved. So that is what is saline solution that we used for cultures, for making culture suspension. So why to use that? Because it is an isotonic solution which provides better environment for cell stability. If water is used directly and it happens that that particular culture suspension is not getting used quickly, it has to be stored, and you have some other work, you have to store that and use it later, then you may find that your culture or your cells, your bacteria, they get lysed. Okay, so we need a isotonic solution, okay, to provide better environment for cell stability. Next is which is the critical step in spread plate or pour plate technique. Okay, so both these techniques are separate. You get same results, but in spread plate, colonies are above your agar surface. In pour plate, colonies are embedded in your agar. That's the only difference. It also depends on your choice whether you find spread plate um, easy or pour plate technique easy for or easy to go. Okay, so in case of spread plate, very critical step is to alcohol gas flaming the glass spreader and then ensuring that the culture is spread all around your plate, not just in between your plate. Okay, if you just spread in between your plate or in the center of your plate, then you will get lots of colonies growing together, as your culture suspension which you had, or the aliquot which you have taken from your culture suspension and you put it on your agar surface, it will not spread properly, and thus all the bacteria will grow in lawn, or in mat or lawn. It will form a lawn in center. In case of pour plate technique, ensuring that the agar butt is not too hot as it will kill our culture, and not too cold as it will get solidified quickly while pouring, right? So in pour plate, what we do, we take aliquot of say, or we pipette out 1 ml of our culture suspension, we put it on our empty sterile petri plate, and then we pour a hot agar butt, not that hot. We test it on our chick to ensure that it is moderately hot, so that it can be poured and media can be swirled, so that the culture suspension gets mixed with the media, and then we keep our plate aside for solidification, right? So that's the procedure for spread plate and pour plate. So there are these two critical steps that you have to follow, otherwise you will not get good results. Okay, then next, now we are moving towards disinfection. So first was inoculation, then spread plate, pour plate, isolation, etc. Now towards disinfection. So why to use 70% IPA for disinfection? So 70% IPA, that is isopropyl alcohol, it is good disinfecting agent which acts on the cell wall proteins of bacteria and it kills them. Uh, then another is, most of the time this question you get in your interviews as well, that is why methanol is not used for disinfection. So methanol has low microbicidal activity as compared to IPA, that is isopropyl alcohol. Okay, bactericidal, that is killing bacteria. Okay, so methanol has low microbicidal activity and is toxic, and it gets easily absorbed through skin and can cause vomiting or decreased level of coordination if you come in contact for a longer period of time with methanol. These are the problems which you can face. Okay, so it is not safe to use in your practicals, as we need IPA for so many times for wiping our working tables, or LF, or biosafety cabinets, or even for flaming our say forceps or glass spreaders, etc. So we don't use methanol, we try isopropyl alcohol. Then yes, even sometimes ethanol is used. Okay, then next is sterility test mandatory for each batch of culture media which is prepared? So what is sterility test here? A simple sterility test which you can follow is pour your plates or make your broth. Autoclaved media is kept for 18 to 24 hours outside, that is in room temperature, and next day just observe whether or do you find any kind of contamination. And yes, if you find contamination in many plates, then it means that autoclaving was not done properly. Okay, so that is the simple sterility test, and yes, it is mandatory. It is done to ensure autoclaving is done properly and there is no contamination in the media. So next is about basic microbiology practices. So first is, what are basic lab practices? which are to be followed whenever you enter the lab. So when you enter your microbiology lab, as a microbiologist, you should follow these basic lab practices. That is, wear your lab coat, that should be cleaned time to time. You should wear lab slippers and not your regular footwears. Why? To avoid outside contamination to enter your lab. Okay. Then wear safety equipments like hand gloves, mask, goggles as and when required. And girls should tie their hair properly. Okay. Very basic lab practices. And this shows that how much you are dedicated to your course and how much you care about your own experiments or your practicals. Okay, because if you don't follow these basic lab practices, then somewhere you will suffer by getting lots of contamination in your media plates. Okay, then next is what are safety practices to be followed while working in the lab. Okay, so first was when you enter your lab, what you should follow. Now what you should follow when you are working in lab. So first is wear lab coat, lab sleepers, safety equipments as and when required. Okay. Then treat all organisms which you handle as pathogens, as you don't know what particular culture is given to you. Whether say for example you are given a simple culture of say E. coli. Okay, you don't know that whether it is pathogenic or not. Okay, and by mistake if you uh, happen to spill that culture or you keep your used glass pipette just like that on your working table, then what can happen? It will lead to an infection. Okay, and someone may suffer, you or someone may suffer from that. So it is better to treat all microorganisms you handle as pathogens. The next is label your plates, flask properly so that uh, someone happens to see what is or which particular plates are these which are kept in incubator, then he or she should know that this particular plate belongs to this particular person who is doing a practical and needs to incubate these plates for some more time. Okay, so labeling is very important when you are working. Even if you have water in your flask, okay, for your any particular experiment, do label your flask as distilled water no matter what. Okay, at least someone who is looking for ethanol will not use it as assuming as it is ethanol. Okay, or someone who is looking for water will not use ethanol which is unlabeled. Okay, so it is better to label your plates and flask, then wash and sterilize your glassware before using for your experiment. So you should ensure that every glassware which you are using is washed properly. Okay, and then you sterilize it, autoclave it, and then use for your experiment. Then disinfect your working table before and after use, and never pipe it by mouth. Try to use micropipettes. Okay, or even for glass pipettes there are some mechanical tools which you can use, but don't pipe it out by mouth. Then never eat or drink in lab, and wash hands properly before after working and before consuming your food or water. So next question is, why are Petri plates kept inverted in incubators? Okay, so in case of antibiotic susceptibility test where you use antibiotic disc, or you bore wells and you add liquid antibiotic solution and you want it to diffuse in your agar, in those cases you don't invert your plates. You invert your plates when you do streaking or spread plate, okay, or even pour plate. But in case of pour plate, let your agar get solidified properly and then you incubate your plates. Okay, so in those cases we need to incubate our plates inverted. Why? To avoid droplets from the lead which are formed due to condensation, and we want to avoid those droplets to fall on the agar surface. Why? Because they can interfere with our results, as wherever water is present, microbes, they get mixed up and they start growing, as you will find water and media present in our media plates, petri plates, then yes, it will just lead to interference in our results. You will not get isolated colony, okay, if you don't invert your plate uh, while incubation. The next is why plates are stacked one upon another while pouring media for media plate preparation. So why we do that, or why to follow this particular practice? So we know that vapor condenses on lid of plate when hot agar is poured. Okay, you pour hot agar and you close your lid of your petri plate. What will happen? In fractions of seconds, you will find that vapors they get condensed. So to remove that particular condensation, what can be done? Another plate of hot media is kept on it, and the condensed water evaporates, and this minimizes the chances of contamination. Okay, and that's the reason when you prepare your media plates, you pour your hot media into sterile petri plates. Try to stack 4 or 5 plates and um, try using this method, stacking plates one upon another. Okay, that's the best method. Then why are media plates kept partially covered when media plates are made in biosafety cabinet or LF? So it means that when you pour hot media in your sterile petri plates, we keep them open, partially open, and we put on the UV lights. Okay, so that particular question is here. So glass petri plates, they can be opaque to UV rays. Okay, so plates are kept partially covered, okay, to sterilize the media while it is getting solidified. Okay, and you ensure that your media plates are getting disinfected again by UV rays. Okay, you are sterilizing it, but while pouring, it may happen that some uh, microbes get settled from air, so we use UV rays, right? Next. Okay, so a basic question, or I will say you should know this basic difference between biosafety cabinet and LF. Okay, so here is the difference given. Question here is, while handling fungal cultures, what should be used, biosafety cabinet or laminar airflow? So the answer to that is, for fungal cultures it is used, or it is recommended to use biosafety cabinets. Okay, good to go. Why? Because airflow is vertical, all air inside biosafety cabinet is filtered and it is circulated. Okay, I am reading this part. So air which is thrown out is also HEPA filtered. Okay, and then it is also burned in case of biosafety cabinets. So there is no chance of outside contamination. And even inside biosafety cabinets, chances of um, contamination by the fungal spores is also less. Why not LF? The airflow is horizontal or in some cases even vertical, but only laminar air is HEPA filtered. Okay, and air that leaves the LF is not filtered and burned, so there are chances that contamination and infection to the handler can happen. Okay, then next is about how a bacterial slide is to be focused. So first you focused under 40x and then under oil immersion lens, that is 100x. Okay, never go to 10x. And if your examiner finds that you are first putting your bacterial slide at 10x and then at 100 or 40x, then you will lose your marks. Then how a fungal slide is to be focused. So now in case of fungal slide, yes, you go to 10x first and then 40x. You don't use oil immersion lens for fungal slides to be focused. Okay, you get a good picture, clear picture under 40x itself. Why? Because fungal cultures, we characterize them as eukaryotes and bacteria are prokaryotes, so they need more magnification. Okay, very basic answer. Then next is, how should uh, how should be the micropipettes kept after using or storing of your micropipettes? Okay, you have done your practicals and now it is time to keep your micropipettes at their place. So suppose you used 100 ml, or sorry, 100 microliters micropipette, and you wanted to pipette out 50 microliters of the solution. So you adjusted your micropipette at 50 microliters. You have used your micropipette, your experiment is done, and now while keeping your micropipette, you cannot keep it at 50 microliters. You have to keep it to its highest volume. Okay, answer is a micropipette should be kept vertically to avoid contamination and dialed to its highest volume. Okay, you can see here, dialed to its highest volume. So for 100 microliters of micropipette, the highest volume is 100 microliters. So you adjust to 100 microliters and keep your micropipette vertically. Then next is why one should clean working platform before and after working. To avoid cross contamination, one should make a habit of disinfecting working platform before working, and to avoid spread of any infection, wipe your platform or disinfect your platform after working as well. Use 70% of isopropyl alcohol or even ethanol is okay. Then next is why to use Bunsen burner with blue flame. Okay, why not yellow or red flame? The blue flame has high temperature on complete combustion as compared to yellow flame, and the blue flame has high temperature, so when you incinerate, or you incinerate your wire loop, it gets red hot in fraction of seconds, right? So that's the reason, and it is the highest temperature which ensures that yes, you incinerated your wire loop, you sterilized your wire loop correctly. Then why Petri plates are labeled on base? Okay, why on base and why not on lid? So the lid of Petri plate can be exchanged easily, so it is better to label Petri plate on the base other than on lid. Okay, suppose you and your friend, you did a very good experiment where you had to streak something, okay, streak a culture, and now you labeled by mistakenly on lid, and your partner also labeled on a lid. If you got your results correctly but your partner doesn't got any results, what can he or she do? He can just exchange the lids of your plates and can show your results as his or her. Okay, so to avoid that, always have a practice of labeling your Petri plate on base. And yes, always label your Petri plates before using your Petri plate. Okay, to avoid the confusion. Or many times it happens that you streak 4 or 5 different types of culture and you have not labeled a single plate. Now you are confused that which plate has which culture. Okay, then next is last question, that is for how many days a sterile and uninoculated plate of culture media can be kept or you can store it. So max for 5 to 6 days a plate can be kept, and it should be stored properly. Okay, not in a very dry or not in direct sunlight. Okay, but should be observed daily for contamination and discard if necessary. Okay, so these are all basic lab practices which I feel a microbiology student should follow when he or she enters microbiology lab. Okay, so I hope this video is helpful to you all. Do like my videos, do share my videos with your friends, and do subscribe to my channel. Okay, thank you for watching. Keep supporting.